Thank you very much, Jamie, and thank you to the team for inviting me to talk to you. Can everyone see the slides okay? Good. So, as Jamie's just mentioned, here's my topic about why is autism more common in males. Before I sort of get into this, a slide of lots of acknowledgements that I'm going to be telling you about research that's multidisciplinary, lots of names on this slide, but they sort of fall into three groups. There's a group of psychologists and people doing neuroscience, mostly brain scanning. There's a second group of people who are looking at the role of hormones and genetics, and it won't surprise you that when we're trying to think of explanations for why a condition might be sex-linked, we have to look at those factors. And then at the bottom there is a group of scientists in Denmark, and I'm hoping by the end of my talk to tell you about a collaboration that we've had, which has been very exciting, between Cambridge and Copenhagen. So here's the basic sort of finding that we need to try and understand, which is autism, this neurodevelopmental condition affecting social and communication skills, and accompanied by these narrow interests, sometimes pejoratively called obsessions, and difficulty in adjusting to change. You can see that basically it seems to be more common in boys than girls. So these are the two major subgroups. Classic autism, where the sex ratio is 4 males to 1 female. Asperger's syndrome, which no longer exists in America, apparently. Still alive and well in Europe if you use ICD, but it's 9 males to 1 female. So we've got a very clear bias that seems to be that if you're male, you've got a higher risk of ending up with one of these diagnoses. And you all know that the difference between those two subgroups rests on IQ, that in classic autism the person might have an IQ anywhere on the scale. And in Asperger's syndrome, IQ is at least average, if not above average. And the other differentiator is language, that in classic autism, the child is invariably delayed in language, whereas in Asperger's syndrome, if you take the history, they talked on time. But those two subgroups on the spectrum seem to be linked to your sex. And this slide, just to keep it very up to date, is a study from the Center for Disease Control in the US. So it came out this year, showing the prevalence of autism across the spectrum, suggesting actually it's getting very common. They estimated, looking at a range of states in the US, that it's now 1 in 48 boys and 1 in 189 girls. So most studies looking at prevalence of autism seem to find it's around 1% of the population. This study is finding it's a little bit higher than that, but we still see the very clear link with sex or gender. It's more common in boys than girls. The graph also shows you the link with IQ, that actually what you can see is that in both males and females, these days, this is like 2014 data, that more cases are being recognized and diagnosed who have no learning disability, so IQ in the average or above average range. So some of you might recognize this man. This is Hans Asperger. So he was the pediatrician working in Vienna in the 1940s. Very interesting time to be working in Vienna. But his name is obviously given to one of those subgroups. And his idea was that autism, as we now refer to it, is simply an extreme of the typical male brain. So that was his sort of anecdotal clinical observation, looking at children in his clinic. And if you just looked at the pattern of behavior, pattern of interests, pattern of cognition, maybe autism isn't anything mysterious, it's just an extreme of the typical sex differences that you see in the general population. So that was his opinion, And obviously, in order to test it, we have to have a notion of what do we mean by a typical male or a typical female brain. So this slide summarizes a lot of developmental evidence from psychologists looking across cultures at hundreds of children during play. So just looking at typical children using a paradigm called spontaneous toy choice, where you put the toys out on the carpet, you video the children, and you code the videos later for which toys did they spontaneously choose to play with as an indicator of what are their interests. And summarizing, you can see that that boys spend more time than girls playing with constructional toys like Lego, and also mechanical toys like little toy cars, on average. Very important caveat. There are always exceptions. And girls spend more time than boys playing with dolls, but particularly creating social stories with their dolls. So imagining that the doll has thoughts and feelings and mental states, again, on average. So this might be suggesting that in the general population, we see one of the areas, or in fact two of the areas of sex difference might be interest in people and their emotional lives, which may be a stronger drive in females, and a fascination with patterns and systems, which might be stronger in males on average. So you could easily criticize those play studies as saying, well, they're not really a reflection of anything except cultural factors, the kinds of toys that the media and the advertising industry targets towards little boys and little girls, maybe gender stereotyping, and maybe nothing more than a reflection of inadvertent stereotyping that's going on by parents or teachers. In order to know whether biology has anything to do with these sex differences, one way to examine this is to look at newborn babies before there's been much opportunity for culture to shape babies' minds. So we did a study in Cambridge at the local maternity hospital looking at newborn babies. They were 24 hours old, and we presented them with two types of stimuli. One was a human face, it's actually a master's student, and the other stimulus was non-social. It was a geometric mechanical object suspended above the crib. And we filmed the babies for how long they looked at each type of object to see if there's any sex difference psychologically that you can detect at birth or in the first day of life. And again, what you can see is that more girls than boys spent longer looking at the human face. This is on day one of life. And more boys than girls spent longer looking at the mechanical geometric design. So this data here just shows you the babies that showed a preference for one kind of stimulus or another. Some babies looked equally long at both types of stimuli. But what we're seeing is the sexes diverging on the first day of life, suggesting that whatever the role of culture later on, it's no doubt there. There might be some prenatal factors, biological factors, that are already predisposing boys and girls to attend to different aspects of the environment and catching their interest. Back to autism. This is a study from California, University of San Diego, by Karen Pierce, showing that at the earliest age you can diagnose autism, which is around 18 months old. If you bring children into the clinic and give them these sorts of stimuli to look at, a social stimulus and a non-social stimulus, and simply measure how long do they look at each type of stimulus. If a child is spending more than 70% of their time looking at the non-social stimulus, the probability that that child has autism is 100%. It's kind of an astonishing finding when I read this, because it's suggesting that maybe we could use a behavioural test for early diagnosis, which would certainly speed things up in terms of doing the assessment. But caveat is that this was only a clinic study. We don't know whether this test would have such good accuracy at distinguishing autism from typical development or other conditions if you used it in the community. Nevertheless, just looking at the pattern of results, it certainly fits Asperger's idea that maybe autism is just an extreme of the typical male pattern of interests, given that we're finding a sex difference at birth or in early infancy, with girls showing a stronger interest in faces and in people. So this is a test that some of you will have come across, the eyes test. And I'm showing you data that's not yet published because we've just done a kind of cull of all of our data from an online platform where adults with autism and typical adults can log in and take the test. So the numbers at the top there, I don't know if they're fitting on, are they slightly going off the edge there? You can see it, are the numbers of participants in each group. So we've got typical males and females, and then men and women with high-functioning autism or Asperger's syndrome. And they're taking this test, which, as you know, you look at the photograph just of the eye region of the face, and you have to pick which of the 4 words around the photo best describes what the person in the photo is thinking or feeling. Here she is meant to be a bit dispirited, a little bit sad, and if you got that right, you did it on the basis of really quite minimal cues of emotional expression around the eyes from rather poor quality black and white photographs. Jamie and I were talking about this earlier, that the fact that we get clear differences even from quite low resolution grey or black and white images is quite striking. But what you can see is that in the typical population there is a clear sex difference. Women score higher than men on this test, and on the left of that bar chart you can see males and females with autism score significantly lower than controls. So they're having trouble reading faces and interpreting eyes in terms of the person's emotional state, but there's no sex difference. This seems to apply both to males and females with autism. We've also used the eyes test whilst people are lying in the scanner. And what's found is that, again, you see a sex difference in the typical population, that women, when they're looking at these images and trying to decode facial expressions, show more activity in left inferior frontal gyrus than men on average. And that's the same region that you find is underactive in people with autism when they're lying in the scanner doing the same task. So again, we've not just seen it at the psychological level, but at the biological level, we're also seeing this pattern that where people with autism might be showing an extreme of the typical male profile. Just as an aside, another way to kind of verify the brain basis of empathy, which is effectively what this test is measuring, or at least cognitive empathy, is to look at lesion patients. And this is showing you 2 studies, one where we looked at patients with lesions in the inferior frontal gyrus, gave them the eyes test, and they struggled with this test. And the other, patients with amygdala lesions. So these are at least 2 different brain regions where if you have a lesion in that part of the brain, it impairs your ability to read faces. This is showing you that performance on the eyes test, this empathy test, is also to some extent genetically mediated, because it's looking at performance in typical males and females as a function of which version of the oxytocin receptor gene they're carrying. So that's a gene that comes in 2 different flavors, and people are carrying either one polymorphism or the other, and depending on which version of the gene you're carrying, it changes your performance, or it is associated with differences of performance on this test. So genetic factors are coming into play, but particularly a gene that mediates a hormone, oxytocin, which we'll talk about a little bit later, a hormone that has been established as playing a role in social behavior. In fact, you can see here that these days research, but not yet clinical, practice is using oxytocin administration, where you can give people a nasal spray and give them an extra dose of oxytocin. And you can see that when typical participants are given either oxytocin or a placebo, and they're blind, so they don't know which one they're getting, when they're experiencing extra levels of oxytocin, their performance on the eyes test increases. So this is suggesting that hormonal factors may be important in social cognition and in empathy, and that this also improves performance in people with autism. There's a related hormone called vasopressin, which seems to have the opposite effect, because when you administer vasopressin to typical individuals whilst they're doing the eyes test, it actually makes your performance worse. And these two hormones, oxytocin and vasopressin, often act antagonistically. Here's a bit more data using the eyes test, showing that genes must be involved in performance in empathy, because it's a family-based study looking at people with autism and their siblings compared to unaffected controls, just looking at performance on the eyes test, showing that the siblings of someone with autism, they perform sort of at an intermediate level compared to their sibling who does have autism versus someone who's completely unaffected. So the sibling is likely to be carrying some of the same genes as the person with autism, even if they don't have a diagnosis themselves. This is some evidence for genetic factors influencing social skills. And the other group, of course, where you can look for genetic factors are the parents of children with autism. So when they're given the eyes test whilst they're lying in the scanner, looking at patterns of activity in the brain whilst they're decoding facial expressions, you find that there is a sex difference, as I mentioned earlier, particularly in left medial temporal gyrus and, in this case, dorsolateral prefrontal cortex. But what you can see is that mothers and fathers of children with autism show even less activity than typical males whilst they're doing the task in the scanner. So the parents who carry the genes for autism seem to be showing, if you like, an increased masculinization of brain activity whilst they're doing the task. The other side of autism is a fascination with non-social stimuli, and the question is whether there's a sex difference in this. So this is the embedded figures test, which many of you will recognize because psychologists have been using it for over half a century, and which shows a very clear sex difference in the population. So the task, as you know, is to find the target shape, that cube, as quickly as you can in the overall design above. If I had a pointer, I would trace it out for you, but hopefully some of you can see the shape. But the data here shows you the average number of seconds it takes to find the target shape, that males are faster than females on average, males taking about 46 seconds, females about 66 seconds. People with autism or Asperger's syndrome are super quick on this test. So colloquially, we would say that they've got an excellent eye for detail. But again, this pattern of performance looks like, or can be interpreted as an extreme of the typical male profile. And once again, if we put them into the scanner whilst they're doing that task, so they're looking for the target shape in the overall design, what you find is that women in the population show more activity in a part of the visual cortex, posterior parietal cortex, compared to typical males. People with autism show even less activity in the same region of the brain. So once again, despite their better performance, they're actually showing an extreme of the typical male pattern of brain activity whilst doing the task. Questionnaires are another way to look at sex differences. So this is showing the autism spectrum quotient, and some of you may have come across this as a measure of autistic traits, which you can administer in the general population. What the two curves on your right show, are people with autism, and the two curves on the left are people without autism. And there's a small but significant sex difference in the general population. So males show a slightly higher score on the AQ, the autism spectrum quotient, than females. And then you can see that the two groups with autism are simply shifted over to the right. So again, we can think of them as showing an extreme of the typical male profile with a much reduced sex difference in autism. And this is the empathy quotient, so another questionnaire that people can fill in as self-report measures. We use it in our clinic, but it's also very useful in research. And what we're seeing here is the higher the score, the better your empathy. That women in the general population tend to score higher than men, so a sex difference in self-reported empathy. And that people with autism, both males and females, are scoring significantly lower. They're shifted way over to the left in this case. But again, it fits this idea of a profile that's the extreme of the typical male one. So we've been thinking about hormones and whether this might be a relevant area to explore. And the easiest way to look at hormones is in saliva or in blood. So you can ask adults with autism to provide a saliva sample or a blood sample, and the relevant hormones to look at are probably that pathway on the left there, which is called the sex steroid pathway. You can see testosterone is at the bottom, but testosterone doesn't come out of nowhere. It's synthesized from these precursor hormones. It starts with progesterone, then it goes to 17-alpha hydroxyprogesterone, then another androgen called androstenedione. We've looked at all of these in blood samples in adults with autism, finding elevated levels of androstenedione, so it's one step in the chain before testosterone, in both males and females with autism. So that's in adulthood, but of course we know that autism or Asperger's syndrome doesn't start in adulthood, it starts in early development, potentially prenatally. So part of our research, which I'll go on to tell you about, is looking at prenatal hormones. But just to show you the role of testosterone, even on that eyes test, this was a study conducted in typical women, showing that if you give them extra testosterone, you can do that by giving them a little patch to put under their tongue so that they get sort of like a male dose of testosterone surging through their brain temporarily, passes out of the system. But you can see that whilst they're on extra testosterone compared to placebo, their performance on the eyes test reduces. So it's suggesting that this hormone does affect brain function, even in adults, and it affects social cognition. But what I wanted to do is spend a bit of time talking about our studies of prenatal testosterone, because it's been known for a long time, about half a century, that this hormone changes your brain development, your brain structure. So it's said to have organizational effects, which is another word for permanent effects, that depending on your level of prenatal testosterone, your brain structure changes and it's basically irreversible. And that's very clearly demonstrated in animals, where you can manipulate the hormone levels and look at brain structure at post-mortem, or you can manipulate the hormone levels prenatally and look at postnatal behavior. Those studies would be unethical in humans, but what we've been doing is looking at humans purely from an observational method by taking advantage of women who are having amniocentesis during pregnancy, so where a needle is introduced into the fluid that surrounds the baby in the womb, that amniotic fluid, and where you can measure testosterone, which is thought to come from the fetus. And we're doing this because many women, as you know, clinically have that procedure, and then they carry on with the pregnancy, so we can look at whether there's any relationship. This is purely correlational, there's no manipulation involved, any relationship between prenatal testosterone and how the child turns out postnatally. The graph on the right shows you that we can indeed measure the hormone in the amniotic fluid. Boys in blue, girls in red. You can see that boys have a much higher level of testosterone, even prenatally. But also notice the huge individual differences. You've got some boys who are so low in testosterone that they're in the female range, but equally you've got some girls who are in the who have high levels of testosterone. They're in the male range. So effectively, we're switching the question. We're no longer asking, is there a sex difference? What we're asking is, depending on your prenatal hormone level, does that predict anything about your later behavior? So we can ignore the child's sex and just look at their hormone levels to see if it's hormones that are driving relevant aspects of behavior. So this graph is kind of schematic, really, just to show you that this is a timeline. You can see birth somewhere in the middle there, and you've got weeks before birth, so that's the pregnancy period. And you can see that in blue is the surge in testosterone in males. So the male fetus shows a spike in production of testosterone prenatally. Girls in red are pretty much flatlining, that they don't really have that big change happening prenatally. And of course, after birth, if we extended this out towards adolescence, we'd see another surge in boys, triggering the onset of puberty. But there's a sort of mini puberty going on in the womb, and people think that prenatal surge in testosterone is all about sexual differentiation of the brain. So whatever differences we find between males and females postnatally, it's due to that surge prenatally. That surge coincides with the time when women tend to have amniocentesis. It's at about 10 to 20 weeks of pregnancy. So by good coincidence, when we're measuring testosterone in the amniotic fluid through amniocentesis, we're likely to catch that surge in production of testosterone. So that just tells you something about the design of the studies. And here, this is again an overview that we're measuring the testosterone prenatally. We're then waiting for the baby to be born, and we've called these children into our lab pretty continuously since then. So we looked at their first birthday, at their eye contact. Their second birthday, we looked at their language levels. When they went to school, we looked at their social skills in the peer group, and also at their autistic traits. And it goes on that we've also looked at their ability to read emotions from the eyes as they've gone into school. These children are part of a longitudinal study. So let me just show you some of that data. So when these children, they're typically developing children, the only reason they're in the study is their mothers had an amnio. But when they were 12 months old, we filmed them whilst they're playing, but this time we're not so much interested in what toy do they play with, but how often do they look up at their mother's face. She's sitting at the side of the room, and she's told to respond naturally to her child, but not to initiate any social interaction. What you can see at the top graph is that girls show more eye contact than boys at 12 months old. So this confirms previous findings that girls show more interest in faces. But down here we can see that eye contact is a function of your prenatal testosterone. The higher your prenatal testosterone, the less eye contact you're making at 12 months old. Obviously, eye contact might be very relevant to understanding autism. Just to help you interpret these graphs, because we'll be seeing a series of them, the red dots are girls and the blue triangles are boys. And the negative correlation here is showing that the higher your prenatal testosterone, the less eye contact you're making. Here is data when the children were 2 years old. Looking at language level, you just ask the parent, how many words does your child say or understand? Give them a checklist of vocabulary. Up at the top, you can see girls have a larger vocabulary than boys. These are typical girls. Again, we've known for a long time that girls talk earlier, their language races ahead more faster than boys on average, that boys are overrepresented in clinics for language disorders like stuttering and so forth. Here we can sort of look at, an association prenatally, because again, we're seeing the line going down. So the higher your prenatal testosterone, the smaller your vocabulary at 2 years old. So when these children were 8, we gave them the eyes test. This is the child version of the eyes test. to see whether there's any relationship with prenatal testosterone. Again, we're seeing this downward slope of the line, that the higher the child's prenatal testosterone, the more difficulty they're having 8 years later at reading faces to decode facial expressions. Here, the correct answer is he's interested in something. Some of you may have got that one right. And when we ask the parents to code their child's empathy, so now they're using the EQ, the empathy quotient, but about their child. Here's some examples of items on the questionnaire. My child gets upset at seeing other people crying or other people in pain. And you say whether you agree or disagree with each statement as a description of your child. Again, what we find is this downwards slope, this negative correlation, that the higher the child's prenatal testosterone, the lower the parent is rating their child's empathy. This is at age 4. Just to state the obvious, but I think it's probably worth stating, is that we've got a sort of double-blind experiment here, because the mother is coding her child's empathy, but she knows nothing about what her child's prenatal testosterone levels were. Equally, we've got a biochemist who's measuring the testosterone, but has no idea what the child's empathy levels later on are going to be. But then we put these two data sets together to see if you can find a correlation. What about attention to detail, something that children with autism are really good at? Here's the children's version of the embedded figures test, where you have to find that triangle as quickly as you can in what looks like a rather Victorian pram, buggy. Some of you may be able to see the triangle. Kids with autism are super quick and super accurate on this test. We've given this to these typically developing children to see if the skill at attention to detail is related to testosterone levels prenatally. So now the line is going upwards. It's positive correlation. The higher your prenatal testosterone, the faster and more accurate you are at finding the target shape in the overall design. And here's a questionnaire called the systemizing quotient. How interested is your child in systems of one kind or another, figuring out how things work or collecting the whole set in a sort of obsessive way, which is all part of normative variation. And what you find is that this is by mother's report, so they're filling the questionnaire in about their child, that a child whose testosterone levels prenatally were higher ends up with a higher score on the systemizing quotient, or how fascinated they are with systems of a variety of kinds. And here's the child AQ. So now the mother is rating their child's autistic traits. So just as we saw the AQ being used by adults earlier, by self-report, so parents can fill in a version about their child. And again, we're seeing the same positive correlation, that if the children who had higher prenatal testosterone are scored 4 years later, this is when they start school, as having more autistic traits. So keep in mind that these are, every dot on the graph is a child that's running around Cambridge where I live. They don't have a diagnosis. Again, they're only in the study, so we could look at this relationship between prenatal testosterone and, in this case, autistic traits. There are about 235 children in this study, which we felt was quite an achievement because it's about collecting the sample before birth, relating it to postnatal behaviour. But of course, if autism is only 1% of the population, 200 or 235 children is way too small a sample to look for clinical autism. There might be 2 or 3 kids in here who have autism. So by itself, we couldn't address the question, does autism entail elevated levels of prenatal testosterone? But what we could do is look to see whether these children, in these children, the hormone is affecting brain structure and brain development. So by the time these children were 8, we judged that they were able to lie still enough in a scanner to look at brain structure. So we know their prenatal testosterone levels, and we've selected one region of the brain, the corpus callosum, the connective tissue between the 2 hemispheres, which shows a very clear sex difference in the general population in the posterior part of the corpus callosum. What we found was that that children who have higher prenatal testosterone levels show more asymmetry in just the posterior part of that brain structure. So this is the first example in humans demonstrating that these prenatal sex steroid hormones affect brain development. It was well known in animals because you can, as I said earlier, manipulate the hormones in rats, for example, and then look at the brain at post-mortem. In humans, this was the first time that this had been found. And we went on to look at the whole brain, so not just one region of the brain, but we did something called VBM, so it's volumetric-based morphometry, where you look at volume of, in this case, grey matter in the brain. You know the children's prenatal hormones, prenatal testosterone, and you're looking for correlations with the hormone in terms of volume of different regions of the brain. And basically what this shows is both parts of the brain where there's a positive correlation with prenatal testosterone or a negative correlation, and there's lots. But I could pick out just a few of those, like the PT up there is the planum temporale, which is a language area, and the superior temporal sulcus, which is known to be associated with looking at the eyes and with making social judgments, theory of mind. Both show correlations with prenatal testosterone. So I wanted to finish with this collaboration with Denmark, and then we can sort of open it up for discussion and questions. That I hope I've conveyed that in our Cambridge sample, we could go a certain way. We could look at individual variation in social skills, language, autistic traits, but in otherwise typically developing children. And that was because of the limited sample size that we had. To look at whether these hormones are associated with clinical autism, we would need a much larger sample size. So then we heard about this biobank in Copenhagen, where the old professor, he's now just retired, since 1980 has been collecting amniotic fluid and keeping it in the freezer. He's got over 100,000 samples from, so it's a national amniocentesis centre. Every time a woman in Denmark has an amnio, the sample gets sent there for analysis, and the professor was very, forward-thinking, thinking to himself, maybe one day these samples will be useful. So we heard about this incredible resource and thought about it in terms of research. So we formed a collaboration with Denmark. So tens of thousands of samples. We've just focused on babies who were born in the 1990s, partly because if you store these samples historically, they decay. So we wanted to stick with relatively recent samples, partly also because diagnosis in the 1990s pretty much resembles how we do diagnosis today. DSM-4 came in 1994. ICD-10 came in around the same time. So what we call autism today is pretty similar to what we called autism in the 90s. So on that basis, we just looked at singletons. That's to say, we excluded twins, because with twins, you're not quite sure whether the amniotic fluid sample belongs to twin A or twin B. So we just focused on singletons, born in the 1990s, and we just focused on males. So we've got almost 20,000 children. The other thing that Denmark has is a central register for diagnosis. This is really handy, and it would be very good if we had this in the UK. So what that means is that every time a child gets diagnosed with autism or any other clinical condition, it gets entered into a central register. So all we had to do was, we know who in Denmark has autism from the whole country. All we had to do is look back to see if their amniotic fluid was in the freezer. And on that basis, we found there were 128 boys with autism or Asperger's syndrome whose amniotic fluid was in the freezer. And of course, we had as many controls as we liked, so we selected twice as many controls as cases. We looked at the whole pathway. We mentioned this one earlier. So we thought, well, here's the opportunity. We won't just look at testosterone. Well, we found lots of clues that testosterone might be relevant. But we'll look at the whole pathway, because if we find testosterone is elevated, we'd like to know, well, how far back does it go, tracing it back to its source, as it were. We also looked at cortisol, because that's not a sex steroid hormone, but it is a steroid hormone in a different pathway. The reason we focused on this pathway, it's called the delta-4 pathway, is because the amount of hormone that you produce as you go down that chain is mediated by two genes, CYP17 and HSD17B2. These are gene names. For those of you who are not familiar with this, every gene has like a number plate, a unique identifier. And these genes have been associated with diagnosis of autism, but also your score on the AQ. So genes involved in this pathway might also be implicating this pathway in autism. In fact, you can see some of those genes here. So I'll skip over that in the interest of time. Here's what the hormone levels look like in the Danish biobank. So in blue are boys, and in red are girls. And across the x-axis, we've got weeks in pregnancy. So just as we saw schematically, there is that surge in production of testosterone, much more pronounced in boys than girls. So it's not the case that girls are completely flat, as we thought. There is a slight surge in girls, and it's all around 12 to 20 weeks of pregnancy. When looking at the 20,000 boys in this sample, you can see they really increase in the amount of testosterone depending on when the mother had their amnio. So that's basically what we're doing, looking at the time of the amnio and the level of testosterone. So here's the main result from that study. We did what's called a principal component analysis. Because each of those hormones depends on the previous one, they're not independent. So you need to look to see if there's a kind of principal component underlying it. So you're throwing all the hormonal data into a statistical analysis. And the first principal component, which we can think of as a steroid factor, shows an elevated level in autism, that's in blue, compared to controls in red. So the elevation is not driven by a single hormone in that pathway, although you can see it here for testosterone. It seems to be occurring right through that pathway. So in pale red or pink on the left are the typical controls. This is just testosterone, and this is showing you across different subgroups on the autism spectrum. So we've got in purple childhood autism or classic autism, in blue Asperger's syndrome, and in green PDD-NOS. You remember that rather clumsy term, pervasive developmental disorder, not otherwise specified. But all of these subgroups on the autism spectrum show an elevation in these prenatal steroid hormones compared to controls. I'm going to finish with just something about females with autism, because in that Danish study we just focused on males. There weren't enough females to analyse. But that'll be a future project to see if the hormone levels are elevated in girls who go on to develop autism, if they're elevated prenatally. But in terms of what we know about women, and this is becoming a sort of a very interesting topic in both clinical practice but also in research, there is evidence for testosterone-related medical conditions or signs in women who later develop autism. So the first bullet point there shows you hirsutism is more common in women with autism, which is another word for excess bodily hair. Irregular menstrual cycle, where women are not showing a regular menstrual cycle, women on the spectrum. Delayed menarche, or age of onset of first periods in puberty, slightly delayed in girls on the spectrum, so they're entering puberty a bit late. And severe acne. All of these are known to be caused by high levels of testosterone. And if you have all of those symptoms as a cluster, you might end up with a diagnosis of PCOS, polycystic ovary syndrome. And again, that diagnosis is more common in women with autism compared to women in the general population. And finally, there's evidence from women on the spectrum that if you ask them about their patterns of interests as children, what toys did they play with, what kinds of activities did they enjoy. They would fit what are called colloquially tomboys, much more interested in the kinds of toys and games that typical boys were interested in when they were young compared to typical girls. And there's this measure called the 2D, 4D, which I'm sure you've heard of. It's the length of your ring finger compared to your index finger. So it's the second to fourth digit ratio, which correlates with prenatal testosterone and prenatal estrogen levels. And in the general population, you can't really see it from just looking with the naked eye, because you have to put your hand on a scanner and sort of measure very precisely, but there's a lower ratio in males than females in the general population. And women with autism tend to have a masculinized 2D, 4D ratio, which might be a sort of proxy for prenatal testosterone levels. So I'm going to finish with conclusions. I think I've shown you that there's various lines of evidence that autism might be an extreme of the typical male profile in very highly specific ways, particularly in relation to the development of empathy, the development of interest in systems, attention to detail, language development, and kind of a limited number of psychological dimensions. Secondly, that fetal steroid hormones like testosterone not only show an association with these typical sex differences in the general population, but are also elevated in children that go on to develop autism. And finally, we do need to extend this research into the neglected subgroup of females on the spectrum. Neglected in research terms because they're often either excluded or only occur in very small numbers in research studies, so we know very little about women on the spectrum, but also neglected, I think, clinically, that whereas we might be quite good at diagnosing and recognizing autism in males, it may be that autism in females more often goes undiagnosed or misdiagnosed. So I'm going to stop there and invite you to visit our website if you want any more information about these studies. Thank you very much.